Okay. Another important way to use histone modification, as we mentioned, these are important, histone marks are important landmarks to tell us whether some regions are interesting. And this is another study done in Eric Lander and Brad Bernstein's lab at Broad Institute. They also look at embryonic stem cells, and they notice that at the promoters of some genes, they see both H3K4 trimethylation. Remember, this is in either active or poised promoters, and H3K27 trimethylation to be enriched on the same gene. H3K27 acetylation is supposed to be suppressing gene expression. And so you wonder, why do you have the activating and the repressive things together? It turns out when they look at all of those genes that have both enrichment and promoter, they notice that when they do the gene ontology analysis, a lot of these genes are very important developmental transcription factors. And they call these bivalent genes because they have both. So you can imagine if you want to control a gene, you can just give it K4. Yes, it will be on or off, just on or not on. Whereas K27 trimethylation is off versus not off, right? Because it only has the off button. But when you have these two together, it actually makes sure that the control is much, much tighter. You want to make sure that you can control it. In case one doesn't work, there's the other. And so having both the on button and off button for the same gene is really a way for the cell to ensure that those genes are tightly and very quantitatively regulated. And so interestingly, so basically later on for those genes, when they need to be activated later, you will remove the H3K27 acetylation mark and only have the K4 mark, and then the gene will have ability to be turned on. But if you need to turn off some genes during differentiation, you say, okay, this now it's going to the blood lineage, it's definitely not going to bone, then those bone region or bone related transcription factors will be silent, and though they will lose the K4 and only have the K27 trimethylation, they will be silent. And so initially having both K4 and K27 is a way for the cell to say, these genes are all important, because the cell at this stage can be differentiated into anything. All of those genes that have potential to decide which way to go, thinking about the epigenetic landscape, they are all being there and getting ready to be on or off as needed. Okay, so bivalent genes really tell you which genes are potentially important. And later on, people also found genes that they're called the super enhancers. And this work was done by Rick Young's lab initially, but the figure is generated from some public data. They noticed, for example, if you look at a transcription factor like a notch, where they bind into the genome, these regions are pretty sharp because remember, transcription factor, they recognize a 6 to 15 base pair region, and they bind there, and you can enrich for a peak that's pretty sharp. But if you look at the histone marks, you can see in here, the transcription factor is binding, and this region is nucleosome free. You can see here, whenever the transcription factor is binding, there is no nucleosome right here, but the nearby nucleosome is very well positioned. That's why you see the bunny here, right? And interestingly, this histone mark, for many transcription factor, the histone mark actually decreased fairly quickly. But in some of these regions, they noticed the H3K27 acetylation signal, as well as things like BRT4, MED1, and these signals not only are tall, they're also very, very wide. And if you think H3K27 acetylation is a proxy for enhancers, those regions that have transcription factor binding but have very tall and very wide K27 acetylation signals, Rick Young at MIT defined this term called super enhancers. They are super, super strong. And when they look at what those, so you can see, you can basically, this is how you assign super enhancers. So most of the enhancers, there's some binding, and you can kind of know that they probably regulate nearby genes, but then these super enhancers might be transcription factors still bind very brief regions, but the K27 signal is much, much sort of stronger, and peaks can be a few kb's long, very, very broad. And when they kind of look at the genes that are near those super enhancers, those genes, they can see, this is, I think, in embryonic stem cell. These, if you look at the genes that really have top K27 enrichment nearby, and there are genes like SOX2, ESRB, NANOG, OCT4, these are very, very well-known genes important for the cell. And so Recon is able to use this to define what are the important genes for that cell condition or the cell lineage. Again, if you look at the expression of these genes, for example, transcription factors, they are not necessarily the strongest expressed genes in the cell using RNA-seq. But if you look at their H3K27 acetylation, you will see that near those genes, there's a huge K27 acetylation. It means that the transcription of those genes are so important. The epigenetic is trying to keep this region really open, really available, and really waving their flags like crazy, allowing other transcription factors to go there and ensure the genes are expressed correctly. And yeah, so these are some of the later publications. It's shown that the master transcription factors in cell identity, usually those genes have super enhancer nearby. And also very often in either cell lineage or even in disease, these super enhancer genes are very important. For example, in cancers, there are some transcription factors that are super important. If you knock out those genes, the cancer cell will die. But how do you know those genes are important? You could look at differential expression. They might be higher expressed, but with this epigenetic indication, if you profile K27 acetylation and you see this gene not only have a high expression, maybe only a little bit, but if it has really high K27 acetylation, that's definitely an important gene because it's a super enhancer gene now. It's more important. Yeah, you can see here, these super enhancer genes, interestingly, are also very sensitive to a particular drug, that this drug really can inhibit super enhancer genes. And when you treat the cancer cell with this drug, those cells will crash and burn, which is really cool. So that's one. And recently, there are also studies showing something called a super promoter, which is also interesting. We show that most of the time when you look at H3K4 trimethylation peaks, they are pretty, focused on the promoter of genes. Maybe only 1 or 2 kb long, those peaks. But then there are some genes, their promoter suddenly have much, much higher K4 trimethylation signal, sometimes can last also kb or 10 kb long, right? So very, very long signals in here. In the normal cells. And these are also indication that these genes are important. These are, in this case, super tumor suppressors. That basically these genes need to be there in order to make sure the cell doesn't go like crazy. They don't grow crazy. And so they have to be expressed to maintain the cell proper function. And so again, having these super promoters will be an epigenetic insurance that this gene is expressed. And when you lose this, you can have cancer, the person can develop cancer. And so again, what they found is that, when you have just a, not necessarily housekeeping, but just like a regular gene, you will have a pretty sharp H3K4 trimethylation. But they found that a lot of these regions that have very, very high and wide K4 trimethylation genes, these are the super promoter genes, and very often those genes are related to tumor suppression functions. Some of those genes are also very, very important for immune cell function. They have to be expressed for this immune cell to maintain its immune identity. And so that's another important finding. Yeah, you can see here.
So I have a quick question.
Yeah.
So it looks like those marks extend past the promoters into the gene body as well. Does it have a function at that point? Because then if it's kind of past the promoter and past the transcription start site, like what's the use in that?
Yeah, I don't think people know this very, very well. First of all, this paper was published, I think, maybe only 3 years ago. And the function for exactly how long it needs to go, it's unknown. And what they can do is they look at all the public ChIP-seq, and because they know whether the sample is a tumor or a cancer or normal, they can look at it, and this is what they observed. So having this mark is not necessarily a direct indication that the mark is important. It's just again, it's like a shadow. Is the shadow important? Well, maybe or maybe not, but at least it gives us some insights on something being quite different between different conditions. And maybe that's a good way for us to prioritize which genes are important for mechanistic investigation. And so this is an example of 2 tumor suppressor genes. You can see in the normal samples, they are very high so that they are expressed to suppress the cell from uncontrollable growth. Whereas in the cancer cell, somehow they are lost, and then the cell will grow like crazy. Right? Then, they see this shortening of K4 trimethylation on some critical genes as another kind of hallmark of cancer, right? Yeah, so you can see in general, all these epigenetic marks are just important flags to tell transcription factor where to go, how much a gene should be transcribed, whether the polymerase should move fast or slow, to make sure, because basically the eukaryote genome, especially for a genome higher eukaryotes, human or mouse, the genome is so huge. When you have a transcription factor to go through 3 billion bases to find where it needs to go to work, it's very, very hard. So having those histone marks really are important landmarks to help the transcription factor find its location very quickly and do its job. Okay, other questions?